Well, welcome to the first Science Cafe of 2026. So excited that you're here with us. Is this anybody's first time? Welcome, welcome. We're glad that you're here. Come back next month. We do this every month. But before we kick off, we do want to say a huge thank you to Last Round for opening up tonight, providing the space for us. So, I get the pleasure tonight of introducing not one, but two special guests. So, Kevin Kertscher is an award-winning filmmaker and communications leader whose career bridges storytelling, science, and social impact. A former pre-med student at the University of Virginia, Kevin ultimately turned to film, earning a master's degree in communications and creative writing from Dartmouth, and has built a 30-plus year contract that spans Hollywood features, PBS documentaries, and nonprofit communications. He's worked extensively with Ken Burns as an editor, and produced and directed the feature documentary Race to the Moon for American Experience. Founder of Big Ocean Studios, Kevin's also led communications for biomedical organizations, including Brown-affiliated Ocean Biomedical and its successor, Secondant Biomedical. Having personally suffered from malaria years ago, Kevin brings a deeply personal perspective to his ongoing documentary while chronicling a groundbreaking discovery in the Jake Kurtis lab and the fight to turn it into a life-saving vaccine and a new class of medicines. And the other half of the duo tonight is his daughter. So Audrey Kertsher is a freshman at Johns Hopkins University, where she's double majoring in molecular and cellular biology and public health with plans for a career in medicine. Do you have free time?
Not much.
Her passion for combating malaria has been shaped by 4 summers working in the Jake Kurtis Malaria Lab at Brown University, where she's progressed from intern to a key member of the drug development team and has helped train incoming graduate students as well. Her research contributions have led to authoring a paper on a promising pediatric malaria drug candidate and presenting the findings at the American Association of Tropical Medicine in 2024. Beyond the lab, she serves as the Johns Hopkins representatives for the University's Ally for Essential Medicines, and self-drafted position paper that they'll be presenting at the North American Conference this spring. She's also an accomplished student athlete, an artist, and is a soccer club goalie, and is active in theater, a former Southeast Massachusetts indoor shot put champion. So, I'd like to hand it right on over to Kevin.
Thank you very much. So nice to see everyone here tonight. I think this crowd is at least 3 times the size that I was expecting, but it's wonderful to see everybody. I make my living with visual communication, and the idea of getting up, when Ann first asked us to do this, getting up and doing a talk without any visuals at all, I thought sounded like a great challenge and very daunting. But what we'll do is we'll try to paint a picture for you as best we can of our view of malaria and the reason we're so passionate about it, and open it up to discussion and let you take the discussion wherever you would like. Neither one of us are scientists yet, and I think Audrey will get there, but both of us have been able to do kind of a deep dive into the world of malaria research and the issues around it. And in that process, we've just been so intrigued by the science around it, by the issues involved with it, by this mountain that thousands of scientists around the world have been trying to climb for decades and are incrementally making progress on it. And I've gotten to follow that. I started in 2019 following a scientist at Brown University who has a major discovery, Jake Kurtis. And then Audrey's had a chance to work 4 different summers in his lab and dove in really deep on the science side. And from that view, we've gotten to learn a lot about the issues around it and a lot about the difficulties of not only making a great scientific discovery, which this lab has done, but then advancing that discovery into a place where it's useful for humans. And that's the struggle that's going on right now, and that part of what Audrey's work there was. there's a number of stories here that we could talk from a number of different angles. And one is typical science story. And when you think of a science story, you probably think of numbers and data and logic and analysis. And that is definitely part of it. And, you know, individual people doing incredible detailed work to advance it. But what I've learned is the other side of the science story, it's also a very emotional story. It's about having a vision and having an absolutely relentless drive to attain that vision, to being passionate about uncovering the truth and being perseverant enough to year after year after year and even decade after decade keep down the road of pursuing that truth, to try to uncover it and do something useful with it. And I think it's a little bit like, you know, a little bit like a ultra marathon run, or, you know, climbing a series of mountains. There's hardship, there's frustration, there's loss, there's great elation and success, followed by incredible disappointment and frustration. And all this, so I've really been struck by the fact that the real science that's happening out there, the best science in the world, is as much a almost heroic journey, a personal journey, an emotional journey as it is a data journey. I'd be happy to talk more about that. This is also an economic story. The economics of malaria, we are all familiar with in some way. We all are aware that almost the whole continent of Africa has lagged behind the rest of the world in development in the 20th century and didn't get to participate in a lot of the advancements that the rest of the world participated in. And a big part of that, the reason for that, I think, is the malaise caused by malaria. It has handicapped whole societies in Africa and continues to. And this is something that Jake Kurtis is really passionate about, that it's not just a matter of saving lives, it's a matter of advancing livelihoods. It's also, I would say, a moral story, and something that we've really seen, especially in the past year, as U.S. aid has been cut. There's an enormous need out there that is not being fulfilled. And basically, the countries that have the ability to fill that need don't have the interest because we're not getting impacted by the crisis as much. And, you know, there's a really big question about whether this issue would still be as prevalent as it is if it were affecting us as much as it's affecting sub-Saharan Africa. So we can talk about that too. I'll give you a couple characters. One character in this story is a 24-year-old filmmaker riding on the back of a truck in Zaire in the late 1980s, heading towards midnight and getting increasingly feverish and shivering, and every 45 minutes or so having climbed down off the truck and run into the jungle to deal with the fact that malaria was onsetting. And eventually the truck driver pulled over and said, okay, I can stop for 2 hours, but then I have to go. And so that 24-year-old filmmaker, namely me, was left behind a tree in a village in the middle of the Ituri rainforest, unable to even, lift my head at the time. And I was woken up in the morning by a group of school children who were kind of poking at me like, like a scene out of a book. And then they got their teacher, and the teacher came, and he knew exactly what to do and gave me what they call fufu, which is local manioc paste, which helped clog up my intestinal tract, gave me papaya, which is really rich in potassium, and tipped that balance back onto the healthy side. But I realize now, knowing what I know about malaria, if that hadn't happened, there's a good chance that it could have gone the other way. there's another character, a 20-year-old brown student who was on a semester abroad in Kenya, and he was riding the night train to Mombasa and got bit by a mosquito and got cerebral malaria and got much more sick than I did, almost died, and rebounded from that. And afterwards took an interest in malaria that became his lifelong passion. And that's Jake Kurtis, who's the principal in this, and worked ever since then to try to understand the disease and figure out a cure for it. And his big question was the question that I've asked myself a lot in this process, which is, all these infectious diseases were conquered in the 20th century. Why not malaria? And so that's something I want to talk a bit about tonight. Another character is a 14-year-old girl, high school student, getting dropped off in front of a tall building in Providence to go to her first day of an internship in a lab, and getting out of the car and not letting her dad even help her check in at the security desk because she said, I got this, no problem. That's a fun story we can go into if you'd like. But the last character is probably the most important in this story, and it's a mother standing over a bed in the Obama Children's Hospital in Kisumu, Kenya, and watching her child slowly die of malaria with her husband standing next to her and holding his hand while he slowly stops breathing. And it's the fourth of her children that she's lost. This is somebody I was reading about this week. And that story happens over and over and over again every day. And just since we've been talking, there's 10 families that have lost children in that way. And that's, to me, that's the most important story in this film we're doing. A little background.
Well, this. Yes.
I know. I'll give you just a couple more facts, and then Audrey will give you the real science. Malaria has been around for about 4,000 years. It was first described in 2700 BC in China, and interestingly, the current medicine that we use now is based on ancient Chinese medicine. And about 40 years ago, the Chinese scientist was working and looking through ancient texts and found references that she thought might have something to do with treating malaria. And out of that, she made the current whole class of artemisinin drugs that we've been using for 40 years, and which are now losing efficacy. Malaria was a huge hindrance when Europeans started sailing out around the world, especially settling in the tropics. It caused all kinds of problems and kept some of that back. In the South, in the U.S., part of the reason that slavery became as prominent as it was because they needed labor that could survive in malaria. And the malaria line in the United States is almost exactly the Mason-Dixon line. And we had malaria in the south of Maryland in the United States until about 1930s, and it was eliminated with DDT. And only because of that, I believe, it dropped off the radar for a lot of scientists, and it was something that wasn't conquered in the 20th century. And the other issue with malaria is that it's an incredibly complex life cycle, which makes it hard to attack. But Audrey will tell you about that.
Yes, so the malaria parasite was discovered in about 1850. The specific strain of Plasmodium falciparum, which is a strain that accounts for about 95% of global malaria deaths, was discovered about a decade or so later. And then in 1897, that's when they finally realized that mosquitoes were the vector leading to the transmission of this disease. As my dad was saying earlier, it is a very, very complex life cycle. So how the basic cycle of a malarial parasite is you get bit by a female Anopheles mosquito. And then that mosquito then transmits parasite to the host, and it settles in your liver. And from the liver, it matures there, and then it moves to your bloodstream, where it starts this cycle of maturing and bursting to release new parasites. The red blood cells burst and release the new parasites, which then go on and infect more and more and more of your body until you are either able to fight off the infection or you die, which unfortunately is the case for a large proportion of the people who are infected with malaria. So I can also get into more specifics about that later. Also, then this cycle continues through the maturing of gametes, which are then picked up by another mosquito bite. And then that is what causes the consistent cycle and spread of malaria throughout a population quite quickly. And even though, that we have malaria nets that are meant to stop these mosquitoes, they are not effective enough to actually stop the spread of this disease. As we were talking about earlier, in the 20th century, we got a much better understanding of many infectious diseases. And this led to the prevention, cure, and eradication of many diseases. Some included chickenpox, diphtheria, measles, mumps, and rubella, and other diseases like that, where we got to fully understand their systems and then go forward and create some prevention types or cures in order to help decrease the amount of people dying from that. Actually, we've seen that in the beginning of the 20th century, the life expectancy in the US was about 47 years. Now it's 78.4 years, due mostly to the development of vaccines and other treatments for these deadly diseases. And I'm sure, as many of you know, malaria is not common in the US. I doubt anyone, goes outside and worries about being bit by a mosquito and getting malaria here. And it's partially because of this that malaria is not seeing large-scale eradication efforts. It is simply seen not as a US issue anymore, except for a very, very small impact in the military abroad. And this is part of what is leading to malaria continuing to kill hundreds of thousands of people every year. In fact, if we look specifically at sub-Saharan Africa, even as the rest of the world was modernizing from the late 20th, early 20th century, malaria continued to harm the population there significantly, and it still does today. So in 2024, globally, there was 282 million malaria cases and 610,000 deaths. Out of this, 95% of the cases and deaths were in sub-Saharan Africa. That math is 265 million cases and 579,000 deaths. And of that 579,000 deaths in sub-Saharan Africa alone, 75% were children under the age of 5 years old. So that's 450,000 deaths. That's over 1,000 children in that region alone dying per day from malaria. And many people think, oh, well, I'm here. I don't have to worry about that. We're not going to get bit by a mosquito. But as climate change is progressing, the area where Plasmodium falciparum, which is again the very deadly strain of malaria, that area is expanding significantly. As mosquitoes thrive in warmer climates, we are seeing, even in the US, we have seen the first homegrown malaria cases since for the first time in about 20 years. So while this may not seem like an issue that we should worry about now, it really is, because this is a global issue that really needs to be talked about. And this is part of why Jake Kurtis, my boss, works on it, because he knows that this is a global issue, even if it doesn't affect us directly here in the US.
So you're probably wondering at this point, all right, so what's going on at this Kurtis lab that's so unique and interesting? And Jake Kurtis, he ended up getting his MD and his PhD from Brown, and he's been teaching at Brown. So he's actually been at Brown since he was like 18 years old, and he's still there. I think he's 56 now. But he went over to Kisumu, Kenya, and the Walter Reed Army Lab has a malaria tropical disease lab in Kenya. They have these in a few places in the world. And they're investing money into trying to figure out how to keep soldiers from getting some of these tropical diseases if they're in tropical zones. And he worked with a guy who's now the head of malaria research at NIH, and they did field studies around Kenya in this basin in western Kenya near Lake Victoria, and also in Tanzania. And everywhere they go, they had a grant from, actually even at that time, from the Gates Foundation, and to take lots of blood samples from kids in villages all around these malaria zones. And Jake started noticing after a while that in almost every village, they would go back and do repeat samples. There were some kids who never got sick. And he asked himself, why is it that some of these kids are getting so sick and some never get sick at all? And he tried to figure it out and did not have success initially. Fast forward about 20 years, the advent of computational biology and a number of other more sophisticated tools made it possible. He had a graduate researcher who was working one night, and they were looking at they were analyzing the proteins that were present in the bloodstream of the kids who didn't get malaria versus the bloodstream of the kids who did. And it was something like 3 million compounds they were going through. And Jake said he was at a party late at night, and this grad student called him up and said, I think you need to come down here. And he found, using these new computational techniques, he found 3 proteins that were consistently present in the bloodstream of the resistant kids that weren't in the other kids. And he had, of course, when he first saw it, he thought maybe it was a mistake. He started looking at other blood samples. They were literally pulling out 25, 30-year-old blood samples out of the freezer and testing them. And he found across the board, these 3 proteins were present. So then they started the hard work of trying to analyze what those proteins were, how they worked. And they zeroed in on one of them. It's called PfGARP, they named it. And PfGARP is present on the outside of infected red blood cells. And they realized really quickly that this discovery had potential to be extremely useful. And
Kids didn't get sick. Did they have malaria in their bloodstream?
yes, and I know you're asking, had they been exposed before? And the answer is yes. And so it was manufactured resistance that some of the kids had an immune reaction from early exposures when they were young that actually created this PfGARP and the antibodies to it, and was in their bloodstream. And so I'll let Audrey tell you how the science started running from that.
Yeah, so PfGARP, it's a parasite protein, as my dad said, that is on the outside of an infected red blood cell. And so basically what was figured out is that when this protein is activated, so when something binds to it, it actually causes the red blood cell to commit apoptosis, which basically means that the red blood cell, it kills itself, therefore killing the parasite that is within it. So what this means is that they found this protein that is a natural kill switch of malaria parasites within the own infected person's body, the own host's body. So basically what I've been doing, what I've been helping with for the last 4 years, is working to develop a new effective treatment that activates this kill switch, because then it would kill all of the infected red blood cells in the host's body, and it would get rid of the malaria. So how this works is we had to work to identify small molecule drug candidates that could mimic the binding to this PfGARP protein. And how this works is basically there's like this big library of drugs that he went through, and he found a handful of them, and we went through and they tested every single one, basically seeing, okay, does this drug kill the parasite? And if the answer was yes, great, and moved on to the next stage. So then you can test that drug in parasites that we know have PfGARP on the surface of them. And we also would test them on knockout strains, we would call them, which are strains that purposely do not have the PfGARP protein. So then we would know, okay, if it kills in the strain with the protein, but not the strain without, we know it is binding to that protein. So that's how we arrived at our favorite drug. We call it drug 19. And we found through a range of tests, including growth inhibition assays and flow cytometry, that we discovered that this drug does activate PfGARP, and it causes these infected red blood cells to die, and it significantly decreases the amount of malaria inside, well, it was in a little vial, but theoretically inside a person's body. In fact, we've gone into tests in vitro in the little tubes and in mouse models, and also this same idea has been extended to a vaccine candidate, not just a drug candidate, and we have seen studies in non-human primates. Specifically, we saw it in vitro, so when we're testing this in cultured malarial parasites in either a plate or in a test tube, we saw that at 47 nanomolar, which it's not very strong, we saw that at that concentration, the parasitemia of these treated cultures was 50% lower than that of the non-treated cultures. And even more, when we tested this in mice, 5 days post-infection, we saw that the untreated mice had a parasitemia as high as 8%. Parasitemia that's greater than 5% is considered severe malaria, so that's a good marker to have. And the treated mice, they never saw parasitemia above 1%, which is amazing. And then even I didn't work so much with the vaccine. I worked mostly with the treatment options. But there was vaccine trials in non-human primates, and we saw that in controls that were not immunized, they had a parasitemia above 8% by day 13 of their infection. So that is very severe malaria. And then in the primates that were treated, by day 13, they had a parasitemia around 2%. And then by day 17, that number had dropped below 1%. And would have kept declining rapidly, but the study was over. So that is kind of the basic findings. And then there are some next steps in order to really work to get this vaccine into human trials and this drug candidate into non-human primate trials. About
Parasitemia? What does that word mean?
Oh, yeah. Parasitemia, it's the percentage of blood cells that are infected with a parasite. So if you have 100 blood cells and the parasitemia is 5%, that means 5 out of those 100 blood cells are infected. Yep.
So these results have been published in Nature 2 times, in Science, and a number of other publications. Jake Kurtis got a major award from the American Society of Tropical Medicine, and you would think that's just about the end of the story, right? Make the medicine. Instead, it has now been 6 years. It's almost like the hard part starts there. It's almost been 6 years that he's been trying to raise the money needed to do human trials. So he's been able to go up to monkey trials for the vaccine, and there's a vaccine, a therapeutic antibody, and a small molecule drug. All 3 are potential useful products. And he's tested 2 of those 3 and all the way up to monkey trials, and he's working on that. Probably in the next 3 or 4 months, they'll be able to do that for the drug. And he was able to get enough money to do that. But so far, he's had no luck yet raising the millions of dollars. It's $10 million to do a human study for any of those 3 candidates. So 30 million total just to get to the first human studies. And that is the game he's been trying to play now for actually over 10 years now. trying to figure out a way to get the funding to cross over from this discovery phase into the real drug development stage. In the meantime, what's going on is they have to test delivery formats. What's the best way to do it? Toxicology, how to minimize toxic effects in advance of the human studies. That's the big deal. Is if there's any toxic effects in the human study, your drug's pretty much done. And so they're trying to optimize for that, and working on the fundraising, and it's been a fascinating journey to observe and to some degree be part of, to watch them doing this. And the only reason they're still going is the passion that's involved and the dedication to somehow figure out a way to get this across the line, to make use of this advancement in science, so that the kids and the families who need these drugs can have them. You want to say anything else for the questions?
I think I'm all set.
All right. We'll leave it there. That's a little longer, I think, than our time was allotted, but apologize on that. But yeah, happy to open up the questions. Any direction? Oh.
So basically, why are they having so much trouble getting funding for this? Really, one of the big things, it kind of goes back to what I was saying earlier, is that well, so much research funding does come from US-based organizations, and many people in the US, basically they don't think that we should care about it because it doesn't impact the US. And this is leading to, this is with malaria along with many other neglected tropical diseases. They have a lot of trouble getting this funding because we do not see a high level of impact in the US specifically. And because of that, people think, oh, it doesn't affect me, why should I care? Which is insane.
And in addition to that, the U.S. is really good at funding discovery science, but purposefully does not fund that next step. And in the university setting, they call it the valley of death, to get across from discovery to implementation. And I've been helping. They've started 2 companies to try to do this. So you basically have to do either pure philanthropy, and it's $10 million a piece on these is a high level for philanthropy, or you do commercial. And the problem is, I've been in meetings with these guys when they present this to people, and they say, how are you going to make money off that? And the way our pharmaceutical industry works right now is that they want blockbuster drugs that can make not millions, not even tens of millions or hundreds of millions, but billions of dollars for years. And that's what the guys who are investing in this, who would stake that 10 million, that's what they want. They want potential to make billions, and they say there's no money in sub-Saharan Africa for it. Yes.
Talking about the United States funding, how's the approach been in something all around the world? It's not just the U.S. or other countries being involved in this too.
Yeah, so the question was, are other countries being involved? Is it just the United States? The United States has really taken the lead, and then in the past 15 years, that's been the Gates Foundation taking the lead. But Great Britain is really active. Oxford has a team that has developed the first vaccine that came out. One interesting thing that's going on right now is that a number of the Gulf nations have gotten very interested in it and want to philanthropically fund and also commercially fund. And now it seems to be happening is crossing over to what they call impact investing, where they can make it an investment in it that has potential to do a lot of good and also has potential to generate return, but maybe not the kind of returns that Wegovy makes or something like that.
Yeah. So the question was, I guess the basis of it was, this new drug, how is it going to attack the life cycle, and will it continue to do this? So I think that what you were referring to, the old drugs, the artemisinin and chloroquine, these were 2 malaria drugs that were very often used in the past, but parasites now are resistant to them. and there really is no way to say for sure that they won't become resistant to a new medicine. That's just, that's very difficult to predict. As far as cysts go, with the life cycle of the malaria parasite, I don't know too much about that. I know more the basic, the liver to the bloodstream and then back out to another mosquito. But I think this specific drug, because it targets all infected blood cells in a host body, that will prevent gametes from forming, and gametes are what the next mosquito can pick up, and that is what matures into the kind of next round of malaria that's given to the next host. And because it's interrupting this red blood cycle phase, it makes it so that these gametes aren't formed, and then that can't be retransmitted from that host. Does that make sense? Okay.
And I'll add on that there's three main stages of the malaria cycle. The liver stage where it's growing, the gametes are growing.
No.
Oh, all right, she told me.
The liver stage is where the initial parasites mature, and then the blood stage, they go through a cycle of ring stage, trophozoites, schizons, and then they burst, and then gametes are a separate part of the blood cycle stage.
I had one instruction tonight: do not talk about schizons. There's liver stage, the blood stage, and the transmission stage. And the Gates Foundation decided about 6 years ago to narrow their focus. They were giving grants to everybody doing everything. Transmission stage is spraying, which is how we eliminated it in the US with DDT. It's bed nets, a number of other things they can do. The blood stage is where the Kurtis lab is working, but the liver stage is where the Gates Foundation decided to focus, and possibly because of those long-term impacts that you get on the liver. But I think, our feeling is that if you break that cycle at any stage, it reduces the number of infections and eventually can eradicate the disease. And that's the hope.
So the question was, with the mosquito, is it only the female, and do we have this mosquito in the US? So yes, the Anopheles mosquito, it is only the females. I actually am not sure if we have them in the US, definitely not in the Northeast. I'm not sure if the recent homegrown cases were, that was in Florida and Texas, I believe.
Maryland.
In Maryland. Thanks.
Are they?
Yeah, I'm not sure the exact biology behind it, but basically so when the parasite bites someone who has malaria, they take up the gametes, and then
Mosquito.
sorry, when the mosquito bites the person, they take up the gametes, and these gametes they reproduce in the gut of the mosquito, and then what it reproduces, that is what is injected back into the person. Do you know about being in the U.S.?
Yeah, I believe it is Anopheles mosquitoes that when it's transmitted in the U.S., that's what it is. But it was DDT that successfully wiped out those mosquitoes. But as what they're seeing now, WHO has put out a big report on this. The malaria, as global warming expands, the malaria zones are moving north again, and so it is expanding because of that, and it's really a race. Is this going to expand before we can get control of it, or can we get control of it before it really proliferates again? And malaria incidents have gone down for about 25 years until 3 years ago, and the last 3 years they've been going up. Cases, infection rates, malaria zones expansion, and deaths have all been going up for 3 years.
It's worldwide.
Yeah.
Yes.
Yes. Oh, interesting. So the question is the malaria parasite a protozoa, and are there other diseases that the technique could be used with? The answer is yes, it is a protozoa.
Yes.
The really interesting thing about this is that the first thing that Jake said is, why does this kill switch exist? Why would, this incredibly, one of the most successfully adaptive organisms in the history of the world, why would it have a mechanism to kill itself as part of its functioning? And his theory on it, and he feels pretty confident about it, is that for any parasitic animal, plant even, to operate, it has to keep its host alive. And so if it overwhelms the host completely, the host dies off, there's no more blood circulating, it dies too. And so what he thinks is that protein has a way of sensing the infection level of the host, and then when it gets to a certain level, it then starts shutting it down so that it doesn't overwhelm the host, and it can live there longer. And I think in that case, it should be able to work with other parasites too. Yeah, Jamie. The question was, can we sort of prompt involvement from Mackenzie Scott by just mentioning her name with malaria and at the same time, and also, is there an avenue right now to potentially donate to this? There is, the Kurtis lab itself does not have a vehicle set up for individual donations. It is something that there's been talk about. And, I think, the biggest thing that I know that Jake would welcome is if anybody has suggestions of somebody who would philanthropically be interested in this, he would love to meet with them and talk to them. There is a group that we've been working with a little bit called the Malaria No More in the UK that's very active and does take donations. And there's another one, United, they used to be called United. I can't remember the name. I could get back to you, sorry. But there is one in DC that's very active on it too, besides the Gates Foundation. But it's a good idea. The question is, are China and India interested in it? India, I don't know exactly how they're doing it, but India is moving towards eradication. And so their health organizations are very active and interested. I think China, they basically are, they're the leaders now in terms of the discovery, but the artemisinin drugs are now, the malaria is getting resistant to the artemisinin drugs that have been working for 40 years. And so they need to be replaced somehow. And I know, China's being very active in biopharma. They're probably working on a number of things, but we don't know of anything that would be competitive with what the Kurtis lab is doing. Yes. So Ann asked about the film that I've been working on. So I started this as a project. I was working for Brown, and they asked me to do a feature piece about this doctor and his discovery. And in the middle of the interview, I sort of sat back. I started thinking, this would be a great documentary, feature-length documentary. And when we finished the interview, I said that to him. I said, this would be really fascinating to follow. And he said, well, I'm going to Kenya in 3 weeks. You should come. So I went home that night and said to Kate, hey, I have a really interesting opportunity. And I hopped on a plane 3 weeks later and went to Kenya. Somehow I got my shots done in time. And then I've been back 5 times with him and watched the evolution of this. In the course of it, they started this company to advance, to get the money and advance it. And I was filming in one of the meetings one day, and they started talking about doing websites and logos and social media and wondering how they were going to put all that together. And somebody said, well, I think Kevin's company does that. So I got pulled in on that. And our hope at the time was that things would advance about 3 years to getting a drug. And instead, and that would have been the end of the film there. Instead, it's been now 6.5 years following and still, swimming upstream with this. And so I've still been filming, I'm gathering footage, and I think it's a fantastic story. And one of these days, I hope we'll find a happy ending. I hope it won't be a sad ending, but at some point, we'll get an ending to it, and then make the film.
Yeah, so the question was, is the type of parasite the same globally, and same with the type of mosquito? I'm not sure about the type of mosquito, but I do know, so there are many different strains of malaria. The one that we in the Kurtis lab are working with is called Plasmodium falciparum, and this is the type of malaria that accounts for about 95% of all malaria deaths. So, and this is also the same one that makes up, I think, the majority, I think it's about 95% of cases in sub-Saharan Africa. And this is the type of malaria, it is not seen in many other places, not to the extent that it's seen in sub-Saharan Africa. And there are many other strains of malaria that are seen in other places like China and India, and even some of the US cases. And those strains are not deadly. So that's most likely there are many, many strains. The majority of them are not deadly, but Plasmodium falciparum is very deadly, and it kills upwards of 600,000 people every year.
The other thing is that most adults can handle a bout with malaria, at least initially, and it's the children. That's why there's children under 5 who their system gets overwhelmed, and they get dehydrated and other issues, and they can't survive it. Adults do get cerebral malaria, can be, that's a different story. In both cases, if you survive though, there's all kinds of long-term impacts. And what I've seen with some of the scientists, we work with a lot of African scientists over in Kenya, and those guys, basically most of them go down about twice a year with malaria. And it's just this recurring thing. And it affects different people differently, but it's kind of like chronic fatigue syndrome a lot of people experience. And there's a number of other long-term effects, even mental illness issues, long-term, that are cataloged as a result of malaria. So it knocks even the 14, 15-year-old kids who can survive it. It knocks them out of being productive students. It hurts people in terms of being productive workers, showing up regularly. It undermines the economy because there's caretakers who have to step out of their regular work routines to take care of people at home. So it really, it's even the survivable malaria that's not as bad has enormous long-term effects. Kate was asking about Seth Letterman, who's a local MD and who started a company and spoke a month or 2 ago. And I heard a lot about his effort. And apparently, so it's $10 million to do the first human studies. And if those are successful, and basically that's a toxicology study to see does it cause harm to people, and if it doesn't, then you do a phase 2 study, which is efficacy. That's about 30 million more at that point. But if you have a successful phase 1 study, a lot of times you can then attract that money for phase 2. And then at that point, if you have a successful phase 2 study, then typically you'll get an offer and get bought out by one of the large pharmas. But then you do phase 3 study, which is a larger efficacy and safety study. And yeah, in the end, that can be $150 million. I think Seth's experience was even higher than that.
He said it's about $1 billion.
$1 billion.
That's what he said.
And you'll see, if you watch in the paper, almost every week you'll see Lilly or Pfizer or one of the other big pharma companies buying out a company for $1 billion or $1.5 billion. Sometimes they're in phase 2, sometimes further. But that's the reason. What the big companies are doing now is they're letting other people take the risks on the early development, and then once they see that there's a clear pathway to make money, then they're jumping in. But Yes, I would like to meet him and introduce him to the team in Providence. Yes. The question is, are there national meetings and are there publications where people are sharing the ideas about malaria? I'll let Audrey answer that.
Yes, so there's one really big conference that I know of, ASTMH, the American Society of Tropical Medicine, and there was so many people presenting about malaria research, and I know there's also even larger conferences globally. You're definitely right, people are definitely very careful about their research, because they don't want someone to take their idea and get credit for it. So that's definitely true. But there are a lot of opportunities for these researchers to share their work. And I went to the 2024 ASTMH conference, and it is encouraging that there is a lot of scientists doing malaria-based work with a handful of different approaches. And it is definitely good to be able to meet with these scientists and compare notes, compare strategies, even if sometimes they are a little cautious when talking to other people researching the same thing.
It's so interesting, the ecosystem, to do this now, the discovery ecosystem is very academic based and it's grant oriented. You get grants from the government, basically, from some nonprofits to do core research, and that tends to be very collaborative, very collegial. Even the Kurtis lab works with about 6 other labs around the country that people are contributing all the time to help advance that science. But because the stakes are so high, once you cross over to that drug development side, people do have to get really protective. Of course, there's patents, so Brown University, the Kurtis Lab, has a number of patents on this intellectual property, and then the companies, when they form the company licenses that patent technology. But the one crazy thing I found out is that in order to maintain patents around the world, you have to pay annual fees. And so you know, South Africa has a fee. Western Africa, a number of countries have gotten together. Southeast Asia, there's different places. India always does theirs by themselves. And so it's about $50,000 to $125,000 a year just to maintain your patents. And if you lose that patent, then at that point, you're not going to get anybody to invest in you. And so possibly if you lose the patent, then somebody can develop it, pick it up and just develop it easily. But most likely what that means is once you lose that, you lose the possibility of making that drug. And having watched this process, I'm sort of interested of either part of this film or a follow-up film would be to look into how many great discoveries have just fallen by the wayside because these incredible scientists are really good at doing their work, but they're not good at the business side of this. And I think it's probably tens of thousands all the time.
Did you ever?
Oh,
Sorry.
well, one sec. Yes.
So the question is, has anyone else identified this protein, PfGARP? It was initially found by Dr. Jake Kurtis. I'm sure that since then other people have realized its existence, but it was initially found by him.
He does have at this point has the global IP on it, except it did lapse in South Africa, and I was in on that discussion. The second question was is PfGARP in everybody? Is it only a small percentage of the population that has the ability to make it? And we think that the answer is everybody has the ability to make it, and it's most likely because it's the parasite that's causing it to be made on the surface of the red blood cell. But one really interesting thing about this is that it appears to be a one-off in nature. This protein doesn't exist anywhere else in any other system. It's just malaria-specific protein. I don't know how they established that, but it's a fascinating piece of which again makes it a really good candidate for for targeting for treatment. The last question, the third question was, is it PfGARP as part of the vaccine? And the answer on that is yes. Did you want to talk about that?
Yeah, sure.
I could feel you.
Sorry. Well, the significant thing is that the idea behind the vaccine and the treatment, it's being able to activate PfGARP. So in each vaccine and each drug candidate that we look at, it's not so much that we are trying to create PfGARP, it's that we are trying to find something that activates it. So another drug that will bind to it and activate that apoptosis mechanism. Yeah. And then we had one more in the back.
Yeah, and so the vaccine and the therapeutic antibody are anti-PfGARP.
That's what we call them.
Yeah. So and so you establish that in somebody who might not have had it naturally.
Yeah. Yes.
The question was this difficult transition that we're calling the valley of death to get from the discovery phase to the human testing phase. Is there anything about the ethical side of that that's really struck us? And do you want to say anything about it?
Oh, sure. Oh, well, I think one thing for me, like the one thing that I've learned working in this lab for the last 4 years is that making a drug or a vaccine that is easily available to any and everyone who needs it is very, very important to Jake, and it's very important to everyone in the lab. Because I know, like, one thing, obviously, a lot of drugs are incredibly expensive, and even though, you know, drugs for all of these diseases exist, they are not easily available to everyone. So one really important thing about this research is that we are committed to making it readily and easily available to everyone that needs it, because that is, a drug can't be effective if everyone can't use it, because then, you know, it's only the ultra wealthy that get to stay healthy, which, no.
Yeah, and I think it's such a fascinating, there's a couple of things going on. One is the money side of it. You know, somebody's at this stage where the discoveries are now, it's about 15 to 18% chance that when it goes to phase 2 or phase 3 testing that it will succeed and become a drug. I personally think that it's a higher chance because of the fact that this is a natural mechanism that they're tapping into. It's not an exterior chemical mechanism that they're doing. But you have to be, you know, the safety issues of actually testing a drug in humans are really important. And, you know, you can't do that carelessly. You certainly don't want to do it carelessly with African children. But at the same time, my feeling watching this up close is that there's something, we've gone too far in the direction of regulatory safety and bureaucratic hoops to jump through. It's making it too expensive. And the drug development advancements that we saw in the 20th century, I think a lot of them would be inhibited now in that environment. And I think there's a better balance that we have to find. I'll let Nate, happy to answer any other questions afterwards.
Awesome. Thank you both so much. Good round of applause.
A little bit different.